Welcome to another installment on the Mariner Biology YouTube channel. This one's going to be about active transport. So we've got to remember that we've already talked about passive transport, and there's two important things to remember about passive transport. One is that it doesn't require any additional energy on the part of the cell. And that's different from active transport, because active transport actually requires that the cell spend some energy reserves to make it happen. Okay, and the other thing we've got to remember about passive transport is that the solutes are going to move, the molecules are going to move from an area of higher concentration to lower concentration. So they're going to move across or down or with the concentration gradient. This is different from active transport, where we're moving against the concentration gradient. Okay, we're going to go from a lower concentration, and we're going to move the solute molecules to a higher concentration. So those are two big differences. Examples of passive transport included osmosis, facilitated diffusion, and just plain old diffusion, where it was the small nonpolar molecules moving straight through the lipid bilayer. Okay, remember with facilitated, it needed the help of the protein, but it was still going with the concentration gradient, and osmosis is moving water from where there's less of it to where there's more of it. So in that case, we're going from what we called a the water is going to go from a hypotonic environment where there's less solute to a hypertonic environment where there's more solute and less water. Alright, so let's get back to active transport. Let's draw out our cell membrane. We've seen this a bunch of times, so we'll draw this out pretty quickly. Got our phospholipid bilayer, and we've got our transport proteins. Alright, let's just think about the cell membrane real quick for a moment. Let's remember that transport proteins aren't the only kind of proteins that we'll see in the cell membrane, but that's what we're focusing on here because we're talking about transport mechanisms. Remember, you've got proteins that help identify cells as ones that are supposed to be in the organism. You've got proteins that act as enzymes. You've got proteins that act as receptors that will hook up with a hormone like insulin. And you've got, I think I got those. I got transport, enzymes, receptors. Oh, and then you have proteins that will actually anchor cells to one another. Okay, but we're concerned with transport proteins here. So let's draw out all the other stuff that we've been drawing with in our previous videos. We've got our concentration gradients, we've got our solute molecules, we've got outside the cell and inside the cell. So we've got the extracellular space and the cytoplasmic space. And we've got an increased concentration of solute outside and a decreased concentration of solute inside. So we're going to say the outside is hypertonic to the inside, or we can say the inside is hypotonic to the outside. Remember, if we have equal amount of solute on both sides of the membrane, then both sides are isotonic to each other. Okay, so if we look at these two green boxes, right, what I'm trying to show you here is that if we look at an equal volume on either side of the membrane, we can see on the outside that we're going to have in that volume more solute and less water. If we look inside in the green box, we're going to see less solute but more water. So the box on the outside has a higher concentration because there's more solute in that given volume. Okay, so now what we need to do is show how active transport is different. So now we're going to take the lower number of molecules and we're going to move those outside to where there's already more of them. And to do that requires energy. And energy comes in the form of ATP. Remember, cells take in sugar, and the sugar is converted into these little molecules called ATP when the cells do cellular respiration in the mitochondria. So what's going to happen is ATP stands for adenosine triphosphate. So there's 3 phosphates on that molecule. We're going to take one of those phosphates with the help of another enzyme and slap it onto that big purple molecule known as the protein. Okay, when we make a molecule bigger, it becomes less stable. And when that molecule becomes less stable, it's going to change its shape. And when it changes its shape, we're going to move molecules from the inside to the outside. We're actually going to pump molecules against their concentration gradient, and that requires energy. So I think that's it. I hope this helps with active transport, and good luck on your test tomorrow. Thanks.